All right, 6 lies in videopolarimetry, how studying evolution helps our Navy. Molly Cummings is going to talk about that. And this doesn't have anything to do with killing little fish and apologizing to them. She has to apologize to the little fish that she takes the brains out of, but not today.
Absolutely. Thank you, Steve. Thanks for the invitation. So, my caveat right away, part of this title was simply to drag you in here and capture your attention, the sex lies part. I do study sexual selection, and I do cut the brains off of female fish to look at their brains, or the heads off to look at their brains. But today I'm going to be talking to you about other parts of my research. I'm an evolutionary biologist, but I don't study fossils, and there's a lot of us out there who study the current tips of the evolutionary tree to understand how evolution, or more specifically the processes of evolution, natural selection and sexual selection, shape animals to be better adapted to their environment or have greater reproductive success. So as an evolutionary biologist, I study basic science questions, and the military funded me for a great deal of money to help them solve human problems, because they recognize that evolution has created solutions to problems that we humans need the answers for. So specifically, one of the things that the Navy needs to deal with is how to hide targets in the middle of the ocean. So those targets could be submarines or mines, or who knows what they're trying to hide. But as our satellites circumnavigate the Earth, they can use 3 different types of light to detect targets against the background. The first one is simply intensity, or the brightness of the light of any given target against the background. That's not very bright on this screen. The second one is the color, or the spectral differences between a target in the background. The fact that these pants are red versus that more gray-green in the background. And the third, which is something that our eyes don't see very well, is the polarization attributes of light. And it refers to the vibrational plane. And I'm going to talk to you a little bit about what polarization is first, but I want to tell you why it's so important. So of these 3 different ways that satellites can detect targets from space in the water, of the 3 ways, polarization differencing is the most sensitive in scattering media. So scattering media is the ocean. So of all those 3 different ways, our technology can detect things best using polarization aspects of light. It's something you and I can't see, but a whole host of animals out there can. And so they've most likely evolved ways to either communicate or hide against this polarized light field. And that's why the Navy has funded me to find out the mechanisms by which they do that. So a little primer on polarization in case you've never really thought about how it works. First, a primer on light waves. Remember, they act both as a particle, a photon, and as a wave. And actually, they come in and all these waves are oscillating in all sorts of different directions. So when the light leaves the sun, the vibrational waves are actually going in every possible direction in the circle. When they hit our atmosphere or any other water-type medium, the specific direction that those waves are going to travel gets biased, okay? And it's usually perpendicular to the axes of the main rays of the propagating light rays. So this is actually part of the reason why we tend to use polarized sunglasses, because as the light rays come in and hit the surface of the water, they're coming off in a very predictable direction. So our polarized sunglasses do a very simple thing. All they do is they block out the light rays coming from that specific direction. And you can test this yourselves the next time you're out on Lake Austin with your polarized sunglasses. All you have to do is turn your head 90 degrees and you'll get a blast of light. The light that's being blocked out from those polarizing filters. Okay, so because light interacts very predictably, or scattered very predictably by water, there are 2 parts on this planet that have very predictable polarized light fields. The one is the sky, because of the water vapors in the atmosphere. And the other is any kind of aquatic environment. So not surprisingly, a number of organisms, from any insect that needs to look at the sky to navigate, to either find home, find pollen, or find where to go lay its eggs, has developed some polarization sensitivity in their eyes. Meanwhile, a vast majority of fish that we know of, at least 5 different families of fish, have evolved visual sensitivity to the polarized light field. So, what I'm going to tell you about are 2 different attributes of the polarized light field that animals have evolved to either communicate with polarized light or to hide against polarized light. And both of these attributes could be used by humans for particular specialized tasks. So I work with a family of fish called Pisciliidae. I don't know if you can see the beautiful male in the top left corner. He's blue. They use a lot of visual ornamentation to attract females. And when we had these guys in the lab, we were looking at them with a videopolarimeter. And this is imagery from a videopolarimeter where the colors you're seeing are not the kind of, we didn't color the background red. Instead, what you're seeing is how the light is vibrating in terms of the angle of polarization. Is it vibrating along the horizontal plane, which is denoted here in red, or some other angle, which would be along the y-axis, either blue or yellow or green. So anytime you see a color pop up on the fish that is not red, it means it's taking that incident polarized light field and modifying it to reflect back a different form of polarization. So you could have very facultative type of signaling going on, that you're being reflected by one type of light and you're reflecting out a different color. So this gave us a clue that just maybe male swordtails are using this polarized light to communicate and be very conspicuous to females. So to test this, we went into the lab. My graduate student Gina Calabrese, and she designed a very specific experimental apparatus to isolate changes in polarization relative to changes in light intensity and color. And it involved diffusion tanks and polarization filters, but the beauty was that at the end of this experiment, we could present females with 2 different types of males, and on one side, the male would be receiving the same amount of light, the same color of light, but he would be illuminated with highly polarized light, and he could reflect his polarized ornamentation. And on the other side, he couldn't reflect his polarization because the light was depolarized. And we would switch males back and forth to make sure we were getting a bias due to polarization. And when she compared whether or not females were attracted to males with their polarized ornamentation relative to when there were no males in the tank, they showed a significant bias to interact with males that were reflecting that polarized light. When we looked further into the system, we noticed that males actually showed significant sexual dimorphism. They had greater amounts of polarization, ornamentation than females. Usually when you see anything that's sexually dimorphic, it means the same trait is different between males and females. That's usually a product of sexual selection. And so we took this a bit further, and we looked at how males and females were using their polarization contrast in social versus asocial environments. And what we noticed is that males, anytime there was another conspecific, another swordtail in the tank, they were increasing their polarization contrast against the background within their bodies, whereas females were not changing it. So what's nifty about that is that polarization could be a very turn-on, turn-off facultative signal that we think is driven by changes in their iridophore structure, which can happen over the order of seconds. And this is something that certainly perhaps the Navy or some other application can use to turn on polarization signals just in specific directions or just in specific times when it's needed to communicate.
Or the what kind of structure?
Iridophores.
Okay.
Yeah.
Explain.
Well, iridophores are, so we're kind of boring when it comes to making color. We just have our skin, which is collagen and a few of these pigmented cells called melanocytes. And fish and frogs and a lot of other classes of vertebrates have many different types of cells that contribute to color that we don't own. So they have not only melanocytes, which make brown and black pigmentation, but they also have erythrocytes that make red and yellow, and iridocytes, which use structural coloration or more reflectance to reflect blues and greens, and in this case, polarization. And at the very end of the talk, I'm going to show you some imagery that looks at those iridophores and how they might be doing. Thank you. And yet, feel free to interrupt at any point in time. So it looks like animals are using their polarization reflectance to possibly communicate or at least attract members of the opposite sex. But going back to the other way that animals might use polarization or reflectance is to try to hide from predators. Now, going back to, let's say, I think 50 years ago, a very famous British scientist named Sir Eric Denham developed a hypothesis that the British military and other militaries began adopting, and that is, how can one hide best in the open ocean environment? Because the open ocean seems incredibly the same, homogeneous. When you look to the right of you, you see one color and brightness. When you look to the left, it seems to be the same. And so he proposed that the ideal way to be cryptic in this kind of environment is just to cover yourself with mirrors. It was called the vertical mirror hypothesis. And this way, no one would be the wiser if you simply reflected back the light coming from this side if you're trying to match the light on your left side of your body. And this was really useful when you only consider intensity and color of the light field. And here's a good demonstration of that. This is a simulation of a fish that's acting like a mirror in the underwater light field, when the underwater light field only has varies in terms of intensity. So it's doing a pretty good job of matching the background light. Not ideally, but not so bad. Okay. However, when you think about polarization attributes, this particular strategy is only going to be useful at one point in time and day, and that is when the sun is directly overhead, because it's only at that point in time when the polarized light field is completely homogeneous in that same plane. On the right side is the same as the left side. And this is depicted up here in the top right, where each disk represents a different angle of polarized light as you look around the fish's axis. And you'll see it's all red, which here represents 0 degrees of angle of polarization. Okay. When you take into consideration that the sun varies, the polarization light field varies throughout the day, so as the sun tilts in the sky, the main plane of polarized light is going to tilt with it. And the result of that is that as the animal tilts on its axis and goes around, it's going to experience very different angles of polarization, and that's depicted up at the top left with different circular disks. And what I'm going to do now is show you a simulation of if an animal is to go ahead and adopt that vertical mirror strategy and act like a mirror, but this time look at the world through polarized lenses, you might notice the animal sticks out quite distinctly, conspicuously against the background light field. So perhaps the evolution has come up with a solution to this problem, and that's why the military gave us some money to identify whether or not animals had indeed solved this problem. So what we did to investigate this was first build a videopolarimeter. And that took a physics postdoc, Dr. Parrish Brady, who received his PhD in astrophysics and came to work with me, a biologist, to look at biomimicry or solutions of polarocripsis in the open ocean. So he built this videopolarimeter, lucky for me, because then we have to go scuba diving with it. And we measured fish and light both in the ocean and the lab. So this is not a typical day for me as a biologist, but a great day for me as a biologist. That's me holding on to one of our oil rig pilings out off the coast of Texas with one hand, and the other hand I'm holding on to Parrish Brady, the constructor of that videopolarimeter. And we're measuring these different fish out around the oil pilings. And we came across a good candidate to start studying polarocripsis because we decided to start with a fish that looked cryptic to our own eyes that were sensitive to intensity and color. And that fish is the lookdown. Maybe some of you are familiar with this animal. It's very skinny, it's compressed, it looks like a mirror, it's shiny. But when we took it into the lab, what we started to notice is it didn't act like a mirror at all. So we brought it into the lab, and we measured it under very controlled polarization conditions that mimicked the natural variation during the day, and it did something very different than a mirror. So this is a lot of physics modeling. You can take all these values, you can estimate what would be the ideal polarization reflectance for that specific environment. And if you want to be ideal in terms of minimizing polarization contrast, you really want to hang out in this purple basin of this optimality contrast map. And what you might notice is there are some white boxes here, or white data points. And all those data points, if they're in white, come from the look down. Meanwhile, we compare different forms of a mirror, as well as different kinds of fish that live in coastal environments and don't have the complex polarized light field as the open ocean. And those measurements ended up further away from the optimality basin. So this suggested, just from the lab, that these open ocean fish were doing something unique from coastal fish to blend into the underwater light field in the open ocean, and better than the current human-made technology, and that is a mirror.
What are the labels on there?
I almost didn't label them. They're different terms of the Mueller matrix, which is a 4x4 matrix that evaluates each component of polarization reflectance. And so what we did is we valued we had a range of values of each of those terms, and then evaluated what the combination of those terms would produce in terms of contrast in the open ocean. So this is just an analytical.
The labels are identical.
Well, what you can't see here is this is a 3. I apologize. This is M33 and then 22. So I apologize. Got cut, squished in the PowerPoint. So optimality modeling is one thing, but the real test is taking this into the field. So what we did the following year is we went into the field into 2 different places where there was where we could have easy access to the open ocean with a nice vertical polarized structured light field. And to do this, we did this in Curacao, which you can have easy access to deep water offshore because it's a volcanic island, as well as the Florida Keys. And here's an example of how we went about, or an example video of one of our data collection rounds. And that is we teamed up with electrical engineers from the City College of New York who built this automated underwater platform rig where we could rotate it 360 degrees, and then we could also vary various different viewing angles of a camera, as well as the fish body position. So we put live fish into or against a mirror, the standard competitor in terms of Crypsis, and set the videopolarimeter, got it going, set down all the angles. We also had devices, sensors to figure out the exact tilt of all the different components, the camera, as well as the target fish. And then we would rotate and take measurements continuously for 360 degrees, and then do this again at different sun angles. So after 10 days of this, we had over 2,000 measurements that accounted for all the different possible angles that predators might be viewing fish and what position the fish might be in. And then we could simply evaluate the relative performance, I'm not sure why this isn't working, of a mirror against the background light, a diffuse mirror, which is just a sandblasted surface of a mirror. I've got a little bit of a problem. And the fish. Okay, there's a diffuse mirror, how it reflects, and then the fish itself. I don't want to get into the details, but they each have a very unique way of reflecting light and reflecting polarized light. And what we wanted to determine is in nature, in the field, which is the best strategy. And so after 2,400 measurements, we can evaluate who does the best job of minimizing contrast against the background light. Fish measurements are in green, mirror measurements are in blue, and anytime you see kind of a gray measurement, it's a diffuse surface. And for not to get into the details, but this F Stokes is a full Stokes. It takes into account all parts of the optical field, and the fish does better than the other. In some of the specific components, it doesn't. However, when you start looking at the exact angles that predators are chasing fish, they come in at very biased angles at 90-degree cone off the tail of a fish. So when you constrain the analysis to those chase angles only, what you find is that the fish does better than the mirror across every single way that you can look at this particular type of contrast.
How statistically significant are those?
P minus 10 to the minus 13. So when you have 2,000 and 2,400 measurements, it's really robust to statistical analysis. So lastly, because this is funded by the Navy, what they want to know is, okay, how do we build an ideal reflector? So the fish doesn't do the best job in every situation. It does it at these situations that has been honed by natural selection, where it's most likely to be running away from a predator. But there are other surfaces, such as the mirror or the diffuse reflector, that can do better at different viewing angles. So what we can do is build a model at what specific combination of viewing angles and target position, what kind of surface reflector would you want? And so that's what we're doing, or in the process of doing. And in parallel to that, we actually have determined the specific properties of the iridophores that you asked about before, and different aspects of the collagen layers, and how they are contributing to this very context-specific polarized reflectance that allows for crypsis in the open. So I wasn't going to get into details, but it's interesting that some of you asked it, but I wanted to put up some of the elements of our data set. But I just wanted to talk real brief, or broadly speaking, that how military applications sometimes have to seek out solutions from nature so that they can develop the absolute best technology for all purposes. So I'd be happy to take any questions. Thank you. Yes?
So to submarine be wrapped in aluminum foil to accomplish that, or?
So aluminum foil acts just like a mirror. In fact, so no. What we're trying to tell the Navy exactly the specific layers of how this fish is pulling this off. And it involves discrete, 2 different layers of iridophores that have specific orientations. And that combined effect allows it to be cryptic over a vast array of orientation, solar conditions. The next stage of this is to determine whether this animal can actually improve its crypsis, and does it improve its crypsis by behavioral modifications? So does it tilt its body to gain even more crypsis? We kept it restrained in generally this position and a few specific positions. But in nature, is it trying to do any specific behavioral modifications to gain even greater? So no, wrapping themselves in aluminum foil at this point wouldn't help them at all, be kind of what they do. But getting very biomimicry of a look down, they could improve their cryptic performance. Mm-hmm.
Do the fish do this when they're dead?
They do, some of this is a passive process, so some of it is simply how they're arranged. Yep, and we can do that, we can compare that from our lab measurements. They get even better crypsis when they're alive, but it's, that's, okay. Anyways, so a good portion of this crypsis is simply a passive attribute of their structures and how they're created and laid out and laid on top of each other. But there might be even an additional element to crypsis that might be mediated by quick changes in iridophores through neurotransmitters. And that's yet another angle that we're trying to pursue.
So if that change that takes place, is that a spatial change, or is that how does that change manifest?
Well, so for one, the passive structures are manifested in that if in one optical plane of the fish, they have a very narrow distribution of their platelet orientation. And this matches very well the vertical distribution of the light field. So if you think about light in the ocean, it's mainly coming from directly above, right? And so their platelets line up really well to deal with the vertical and downward feature of the light field. If you look at the platelets that are important for reflecting light in the horizontal plane, those have a very wide distribution, and that wide distribution helps to randomize the polarized state of the reflectance, and that helps with the crypsis part. If they didn't randomize it, they become much more conspicuous, and so that's one way that they deal with the change. Now you're asking how could they...
Yeah, what exactly is changing? Is it they're changing like molecular spacing or?
So these are these guanine crystal platelets. So they're changing the orientation. Angle, yeah, sorry, yeah. So right now, we think that they're physically just stuck in a given position. They can, some of these guanine, some fish iridophores have the ability to be motile, to move by activation through nerves. So that we have not provided any data for yet. We have hints that that's going on, but we haven't quantified this. This is simply, after you've killed the animal, looking at the structure, what do these different layers tell us? This slide is. Yes?
Can we build iridophores yet?
Oh, yeah. So I go and report to the Navy, and they are happy to have this kind of data. Yeah. It's just guanine. So, yep.
I think there was talk about experimenting on squids and octopuses.
I have a colleague who's part of this project who does exactly that, who works with squid and octopi. And he's been focusing mainly on their chromatophores, which are more responsible for the oranges and the yellows, and less on the iridophores and the polarization. When we were down in Curacao, we were fortunate enough to capture some video footage of a few of the local squid species. They were clearly not being cryptic though, they were communicating with each other, but they were communicating with each other apparently with some polarization signatures. But we haven't yet started to examine squid polarocripsis. But it would be great because that's a parallel or convergent pattern from evolution in terms of how they might be mediating these processes.
Is this different for fish that are closer to the Arctic where the descent angles are different than?
That's a great question. So I would predict that fish that live way down there, where they actually have they're really constrained with the solar angles, they should, you're right, have a different optical axis bias than fish like these, who are stuck with the full range of the solar angles that go throughout the day. So it'd be actually fun, that's a great comparative and test of this hypothesis, to look down at the poles where you have more constrained solar angles that the fish are exposed to see if their optical axes via structural setup of their iridophores varies accordingly. Yeah. Yes?
What is about the structure of the eye that allows some fish to see the polarization as opposed to our
The $1 million, okay. Ay, caramba. Alrighty, so, okay, backing up. Right now, no one knows. And there's a ton, it's a really exciting time to be in sensory ecology because of this. Because to think that we don't understand how vertebrates, other than ourselves, can see a sensory modality is pretty wild to be in this century and still not know the exact mechanism. We have a better understanding of how insects see polarized light field because their visual pigments in their ommatidia are arranged spatially so they can be at orthogonal angles to each other. And you need kind of spatially distinct detectors to pick up polarized light. Vertebrate photoreceptors, in general, we all have our photoreceptors pointing in parallel with each other. They're hanging out, actually pointing in the back of our heads, but they're all in parallel. So So people have wondered, how the heck are other species, invertebrates, picking this up? So there's a couple of hypotheses. First of all, the anchovy has already evolved a set of receptors that do orthogonally, are spatially orthogonal, and they pick up polarization that way. They seem to be a unique branch in terms of polarization sensitivity. One hypothesis, which is pretty wild and pretty crazy and difficult to test, has to do with the fact that if you think about all the vertebrates who can see polarized light, all those other vertebrates have a type of set of photoreceptors called paired or twin photoreceptors. And this is generally in the green photoreceptor class, and it's when they share an inner segment and they have two outer segments coming out. Mammals don't have this. And the reason why, what's been proposed that all the other classes of vertebrates can see polarized light is maybe it's something about these paired photoreceptors. And here's the crazy part. So they're like this. And the idea is that light that bounces off one hits the other and is absorbed. And it bounces off the other one and goes in another direction and is absorbed over here by a different photoreceptor. So the photoreceptor not only acts as an absorber, but a reflector of polarized light. And it's a wild idea, and it's really, I don't think anyone's been able to test it. But yes, we do not know how we can see polarized light. But if you want to see polarized light, we have vestigial capabilities to see it. So if you have a, back in the day when we used to have, old SLR cameras and we put a polarizing filter on the outside, any of you still have a polarizing filter? Awesome. So what you can do, and today's a pretty good day to do it, is go outside and look about 90, find a cloud that's roughly 90 degrees from the sun. So I don't know where the sun is right now, but let's say it's over there. And find a cloud that's 90 degrees from that. And then take your polarizing filter, and because you need aid, because we can't see it with the naked eye. And you put it up and look, just relax your eye and look at that cloud. And in the center, real faintly, you will see a cross. And one arm of the cross will be blue, and one arm will be yellow. And these are called Haydinger's brushes, and they're named after an Austrian or a German mineralogist who identified these when he was using polarizing filters to look at minerals. And then what you can do is take your polarizing filter and then turn it 90 degrees. And those colors of those arms will switch in your eye. So no one understands really why, and we only see it in our fovea, which is the very center of our eye, why we can still see this or what it means. But it's a great byproduct of evolution. It appears to be left over from the kind of visual systems we inherited from our other vertebrate classes who can see it quite well.
Thank you again.